All right, thank you guys for coming. I know it's on a different day, so appreciate you guys showing up. Our presenter today is Dr. Emmanuel from University of Maine. He got his bachelor's in math, physics, and a minor in atmospheric science, and a master's in oceanography from the University in Jerusalem. He got his PhD at the University of Washington in 1997, studying the instability of fronts. He completed a year of postdoc at the University of Washington with M.J. Perry, studying marine optics. Moved to a second postdoc with Ron Zenveld at Oregon State University. He stayed there as a research faculty for 3.5 years before moving to a tenure track position at the University of Maine, and he's been there since 2002.
Thank you very much. So what I'm going to talk to you today about is work I've been doing for probably the last decade, a little more. It has to do with trying to instrument profiling floats with other sensors than what usually people instrument them with, and talk to you a bit of what we learned in the process. So what is an autonomous float? The advertisement, you saw this picture, which is me with a lot of equipment on my back. This is not an autonomous. This is a human seesaw, maybe. And all the blue, white stuff is buoyancy because of the weight of the equipment I have on my back. But this is what we call a profiling float. And just to introduce you to what how it works and how this, we have here a movie that was done by people much more professional than I am. So about 2 minutes.
Scientists on research ships can sample the ocean only briefly, so they deploy automated devices for long-term ocean monitoring. This float is part of an international research program called ARGO. It measures ocean conditions that drive events such as El Niños and climate change. Like a hot air balloon, it drifts along on slow-moving currents in the deep sea. The float dives by mechanically decreasing its volume in the water, and it descends to as far as 2 kilometers, more than a mile, to survey otherwise unmeasured
and this is the CTD.
underwater
Usually
regions.
they only have CTDs on.
Submerged for up to 10 days, the float can be programmed to stay at one depth or to move up and down to follow changing conditions. At preset intervals, it ascends by increasing its volume. Rising through many ocean layers, it records the ocean pressure, temperature, and salinity, collecting an up-to-date profile of evolving ocean conditions. At the surface, the float makes radio contact. Its new location reveals the features of underwater currents, and the data supply real-time measurements of subsurface ocean conditions, details unavailable without ARGO.
It's not really
After
radio,
a
but
short
that's
period,
okay
the
too.
float is ready for another cycle.
Most of them are now Iridium. They use the Iridium network to send data.
With 3,000 floats in a global network, the ARGO program will supply real-time ocean data for immediate use in research and operational forecasting of marine and climate conditions worldwide. ARGO, observing the oceans in real time.
So I would argue that this is our real, the real observing system we have for the world ocean is now ARGO. We did this one. We have really nothing else that covers as globally as this. So this is as of yesterday, about 3,600 floats going up and down. And all of them have CTDs. It's an international system, so every country puts a certain number of floats per year, and they coordinate everything that has to do with the data, but every country can offer as many as they want. And there's actually this week in Ottawa, there's a group that sits down and argue about the definition of the data, names, how do you correct for certain spikiness in salinity, how do you treat new sensors like the ones I've described today in terms of adding them to databases. The float, as it comes up, the data goes straight to 2 databases, one in Monterey, one in France, and is available for people to do modeling of future conditions in the ocean within a few hours. And then there's real-time data, and then there's whole procedure to correct for problems in data that's delayed mode. And there's a lot of, it takes a lot of people to make the data as good as they are, but it's an amazing system in terms of, as a model for international collaboration. and still allowing people to do science with them. So you can have your float be part of it, but you can have a different cycle, for example, either going to different depth or profiling more than once every 10 days. The large majority of them have only CTDs, but there's and about less of them, about less than 50 with biooptics. I'll talk at the end about a new programs which will increase that by at least a factor of 2 that we're now involved in. So what's the limitation? It's currently funded to measure only CTD in the US. In France, now 20% of their floats have additional sensors than that. There's quite a few that have oxygens on them, several hundreds. And the limiting part of the integration was both culture, meaning the people that were running, and it started by physical oceanographer, and they were somewhat reticent of the power consumption and the possibility of problems to the rest of the float if there's failure with these other sensors. It also was a problem that we didn't have a lot of robust sensors that we could count on to measure other things that have been as tested as CTDs. But they had problems with CTDs in the beginning as well. It's not, and some of you might remember there was a Nature paper that had to be retracted because they claimed some warming that wasn't there. Okay, so what I do is optics, and I'd like to introduce you a bit to what are the things I'm going to be talking about and how we use them to study processes in the ocean. So I'll start with absorption. I have here, I put food coloring into three little buckets, or whatever, containers, and I have 3 LEDs. And every talk I give, I do demos. I learned that this is the only thing you'll remember a few months from now, I promise you. You'll see me in a meeting and say, aren't you the guy who did this? This is the only thing you'll remember, nothing about what I talked, but it's okay. It's at least something. So I have 3 LEDs. There's a red, there's a blue, and there's a green. And I have water that matches. And the question I'll ask you is, which one do you think the blue is gonna penetrate through? Would it be the blue, the green, or the red? And when I ask kids that, it's about 50-50 whether it's gonna get absorbed by the blue or it's gonna get through the blue. What do you think? It's okay to be wrong, you know, it's all the fun. Is it gonna go through the blue or get absorbed by the blue? Some said through, some said absorbed. This is what I want. Okay, so now we need to know. Okay, so here I'll put it on the blue. So what? Yeah, it's absorbed, or yes, it goes through? It goes through. The green goes through the green, the red goes through the red. So what does it tell us? It tells the color we see is a color that's not absorbed. So the green grass is green because chlorophyll does not absorb in the green. It absorbs in the blue. And a little bit in the red. But so what we see is what's not absorbed. So this is one. Second, in order for us to see it from space or to see it back in an instrument, it needs to come back to us. And that process is called scattering. So I'm now gonna, so scattering is the process by which photons, I'll do this one, it's gonna make a bigger effect. Photons come to eye. Can you see the beam? The beam, not the point, the beam. No. So all these, I have a good friend that says, all these science fiction movies, they lie to you. You'll never see the laser shot in space. You cannot. I mean, now let's see water. Can we see it in water? No. Again, you see the sides. So in order to see it, we have to add some scattering some scatterers into the medium. So this is Maalox. It works great. You can use milk too. Maalox is an anti-acid you can buy here for. Okay, so now, can you see it now? No, I don't see the beam. No. Can you see the beam? So the only way you can see something is if it's scattered to your eye. So it has to not get absorbed and then come back to you. Okay, so 2 things influence ocean color, which is not the topic of my talk today, but in order for us to see color from space, it's the color we see of the ocean is what's not absorbed and then backscattered. With an instrument, with a scattering instrument, it's what's not absorbed and come back to the instrument. Now, the process that affect how much light makes it to that point, there's 2 processes. There's absorption along the path, and there's scattering that takes it away. And this is what we call the beam attenuation. And people do measure that as well in the ocean. So how much light makes it there? From this we calculate transmission, and we invert that to beam attenuation, which is the sum of absorption and scattering. Now we have another property that is very useful for us, and that's fluorescence. So I have here an extract of spinach leaves, and it has this pigment that phytoplankton have as well as plants. It's called chlorophyll. And what's special about chlorophyll is it absorbs in the blue, and then it re-radiates in the red. So I hope you can see that. Do you see the red dot? So the red dot is the fluorescence. It doesn't come from my laser pen because my laser pen is in the blue, but it gets absorbed and then re-emitted in red. And this is how on these floats we have this fluorometer. They send blue light into the water and they measure red coming back. And they have a filter in order not to measure the scattered light. They only measure what's fluoresced by phytoplankton. And of course, these proxies are not simple. You get different scattering depending on the size of the particle. Physiology of phytoplankton affects how much is fluoresced for a given phytoplankton and species affected. But overall, we find it very useful. And the reason we find it useful, I'll get to in a moment. So this is true for acoustics. Just everything I say now is true for acoustic, although I don't know of inelastic scattering in acoustic, but I'm sure there's some example for that as well. The problem that's well posed is if you tell me the particles and the wavelength of light that you're shining on them, I can tell them how much they scatter. That's an easy thing to do. You put it into, there's codes for it, there's even an analytical solution. What's hard is I measure scattering, or I measure fluorescence, and now I want to know what's in the water. And it's ill-posed in the sense that there's the possibility of having multiple solutions. I can have different combination of particles that will give me exactly the same response. So this is why we call it ill-posed. So if it's so complicated, why do we insist on using it? Well, there's several reasons. One is we're very lucky in the oceanography. We have a huge dynamic range in concentration of stuff in the water, either between surface and depth, between seasons, between when it's stormy and when it's calm. And so it's still very useful, even though we can have large uncertainties in these inversions of the order of a factor or two, and we can tolerate those. Because phytoplankton in the surface ocean can change from 0.01 to 10 microgram at the same location through the year. So it's a factor of 1,000. So a factor of 2 is not a big deal. It's not like, it's not temperature, it's not salinity. These are really big changes. The other thing that these sensors allow us to do is to equip many platforms with them. We can be there, we can have them measuring in the ocean when our boat is not there. And the biggest problem of oceanography is undersampling. The ocean are 70% of the globe, and how many research vessels do we have at any given time out there? Almost nothing. So by being able to do these type of measurements in the ocean, we keep a presence that is useful for us to see how much biased we are by just going with boats that tend to stay not too far from the coast. So what do we use optical proxies for? We use them for temperature, and that's for years from space, near II radiance. Nitrate and sulfides, that has been worked on for the past 10 years. Nitrate sensors are now on floats. Ken Johnson has developed a sensor, it's commercially available. He just added another one, which is not optics, but it's pH, and it's important for some of you, so it's good to know. DOM, hydrocarbons, oils, fluorescence, excited in the UV and measured in the visible. So the same kind of fluorescence I showed you, just with different excitation emission bands. Absorption can be used as well. If you have absorption by dissolved substance, so you have to pass the water through a filter and then measure absorption. Particulate matter, particular organic carbon, for years we've been doing it with attenuation, scattering, ocean color. Phytoplankton pigments, they fluoresce. They have very specific absorption properties, and as you know, we can map chlorophyll from space. Size distribution, there's one proxy is looking at the spectrum of the particular attenuation, how it changes, tells us information about size. Forward scattering, those of you who have heard about the list, it measures how side is scattered at different angles in the near forward and invert that to get a detailed side distribution. And again, ocean color, recently people have started claiming that they can do seismos. It's still work in progress. And finally, we can even do composition. If we look at the backscattering to total scattering, or the degree of polarization in the water, we can say something about whether we're dealing with organic material or inorganic material. And that's great, because the more we advance in this direction, the better we can delineate what particles we're dealing with in a given environment. Now here is an example. This is from the Alliance of Coastal Technology. It's a NOAA funded group which looks at different instrumentation and see how well they work to measure a specific property. And here they looked at an intercomparison of turbidity sensors and different methods to do turbidity. And optics has been used for years and years to do turbidity, either attenuation, side scattering, or back scattering. And what I've done in this paper is just take all the data they collected. I did not collect any one of it, and just plotted the optical property, be it, and this one doesn't work, be it beam attenuation. Again, that's the one where we're looking at how much light makes it to the corner. This is beam attenuation versus concentration of particulate material. This is back scattering, similarly, and this is side scattering. And this is on a linear scale, this is on a log scale. You can see that there's one group that stands out. This comes from Hawaii. And it stands out because calcium carbonate is very, very, very special optically. It has a different index of refraction in this direction and this direction. So it does not line up well with the rest of them. But once you remove them, you can do very well so 95% of the cases, your model versus what you measure in situ is within a factor of 2 if it's beam attenuation, less than that if it's back scattering, and again a factor of 2 if it's side scattering. So it's worked pretty well. Again, a factor of 2 is good enough for you. But it also indicates that if we had some way to measure these, say with 2 polarization, we might be able to separate calcium carbonate in the water. And indeed, Jim Bishop has done just that. He has a sensor that is still not commercial, but that looks at calcium carbonate coccolithophores in the water using this kind of method. You're looking at polarization. Okay, so a little bit of history of the work that consists of putting optics on profiling floats. The first one was Greg Mitchell of Scripps with Matti Karu and Sherman. They used one of the solo floats from Scripps, and they had radiometers. So radiometers just measure light at different depth. And if you look at the derivative of the light with depth, it's the attenuation coefficient, diffuse attenuation coefficient, and it gives you information about chlorophyll and dissolved substance in the water. And indeed, this is temperature here. This is chlorophyll. You can see surface chlorophyll expression in a spring that then goes down to deep, and it was consistent with satellites. The next set of these solo floats was work that Jim Bishop did with Russ Davis and Sherman. Again, at Scripps, although Jim at the time was in another place, he's still in other places in Berkeley now. And what they did is instrument it with beam attenuation meter. And what's very special about that work is they have a transmissometer. So there's 2 windows and the light beam. And what Jim did observe is that when you park it at depth at 1,000 meters for a few days, stuff falls on the window. And your reading before the float comes out is significantly lower than what it is after the float goes up, as water cleans the window. And that's a direct indication of settling by particles, which is this carbon flux we're looking for. So recently, a student of mine analyzed data we collected, again with transmissometer, but we had a much finer resolution of measurements down at depth. Every hour we did a measurement. And she was able to see the flux of particles going down every hour, and sometimes events where stuff washes them off, and use that as a proxy for carbon flux, comparing it to nearby sediment traps. So it's a very interesting method that a lot of work will be done with, I hope, in the near future.
How do you determine that it's actually calcium carbonate and not?
Oh, this was not calcium carbonate. What I talked about was not calcium carbonate, just stuff falling on the windows.
Oh, so
his calcium carbonate stuff that he has, so this is his other sensor, is looking at light shined. So you have a laser in a specific polarization, and you look at the response in the opposite polarization.
I understand that. I thought you were saying that this experiment with the transmissometer And the material on the mirror was actually calcium
No,
carbonate.
no. That material is just stuff
It's
that's,
snow.
it's marine snow.
Yeah.
Marine snow, whatever it is that's produced at the surface and falls down. It can be fecal pellets,
Yeah,
whatever.
yeah, exactly.
What's the settling velocity of these particles that you're studying?
We don't know. I mean, the float is sparked. It's neutrally buoyant, and its stuff accumulates at a specific time. So you're right. What you're measuring is how much stuff collects on it. It's a function of the settling velocity and concentration.
So
Because it's concentration per time.
Water is just flowing through the volume.
Yeah, so it goes, so water flows from the side, but hopefully the float mostly moves, so you have it's here, you have your collection, and the float moves with the stuff, and then particles, and you have to calculate something about the shadowing due to the float, because a particle from this side is not going to come on. And then stuff falls and makes its way to this. And this is why it's a proxy, because there's a lot of things, it has to, there's a top can and a bottom can, and somehow the particle has to be able to come. So there has to be shear in the water column to make it settle on it. But when you look at that and you correlate it with nearby sediment traps, you'll find it where there's a lot of stuff falling down, there's a lot of stuff falling here too, and then there isn't there as well. So it's a proxy at the moment.
Shear or pressure?
Pressure in what sense?
Pressure gradient, you would get it also. You wouldn't necessarily need shear.
Horizontal pressure gradient.
Yeah.
Yes. I mean, something has to, but that pressure gradient can move the float just as well. So
Certainly,
yeah. So there has to be some
But
relative
But if
motion.
you're in the middle of an eddy, then you have a negative pressure region
Mhm.
that's shed.
Okay, I'd love to talk about it maybe afterwards. So another thing that Jim did is in places where they did iron seeding, he had floats both outside, which are the control, and inside the area where they seeded, and again, found much more material settling in the area that goes. And finally, this is the work that we started. A very small POC from wet labs, had backscattering and fluorescence. That's all we're measuring. And the idea was, again, to come up with some sort of a commercial sensor people can start and think about in the large context of the Argo for long deployments, because all of the Bishop deployment to that time were short, very specific experiments. We got some seed money from NASA, and with work with Steve Reiser's group at University of Washington. This is Dana Swift, who was absolutely phenomenal. It's one of the unsung heroes of oceanography. This guy helped the whole community with Argo floats and solve problem and write software and share it with everybody. So we lost 4 sensors in the process. It was supposed to be one float with one sensor. We lost 4 sensors and we lost a float in the process. So it cost much more than $50,000. But this is what happens when you try something new. The float survived for more than 2 years, 221 profiles. It started here, stayed for 2 years in the Labrador between Greenland and Newfoundland, and then crossed here the North Atlantic current, the extension of the Gulf Stream, and died further down. So these floats are expendable. Put them in view, which is one of the features associated with them. This is chlorophyll at the surface, at the top, compared to MODIS. MODIS is in the black. This is chlorophyll based on the float. And we made them fit by doing a linear fit between them. Same goes with, but this is backscattering. We did the same here. So over the whole life, we made them agree. But this is to get the slope. And this is down the measurement at near 1,000, between 950 meter and 1,000 meter. So what you saw is that there is no drift in the sensors. Sensors are stable over the 3 years they've been in the water. There is an increase here in backscattering that comes back down, which is associated with an eddy event in the water that's barely observable in the satellite record. And that eddy event doesn't have chlorophyll in it, so it's not this thing. In backscattering, you don't see it. And these are the measurements in the water column. And so what I'll describe now is a bit what we learned from it. So first, there's this eddy event. We find a very nice correlation with satellites. So which is great because it tells us both that the satellites around the float measure something very similar than the float measured. So it gives us confidence in both of them. The second thing is this huge eddy here, in which in November there's no bloom. These are the periods of growth of phytoplankton. Suddenly we see it. We still have no idea to this day what it is. If you look at surface altimetry, the float was in. The size of these dots has to do with how much concentration of material there was in it. Very large quantities. This is the trajectory in the water. About 2 months, and the float moved with this eddy. It trapped a lot of particle, and the particle extended down to 1,000 meters. To this day, we have no idea what it is. Some people suggest it might be a bloom of Yaxlei, but in order for this to be true, you need it to be coherent top to bottom for 2 months. And I don't know what mechanism could do it. Maybe lateral input, but that's not consistent with temperature and salinity properties. Concentration by the eddy, I don't know how the eddy can concentrate particles. In aerosol deposition, we looked at maps of transmission through the atmosphere. We don't see anything anomalous there. So, still not know. But if you look at records in bats of sediment transferred to depth, so sediment collected at depth, what you find is often around December, they found these big deposition of particles. Why they make it to 3,000 meters around December and not other time, it's not obvious. But they get these large events in between years, here, 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 here. So there are times in the winter where stuff makes it deeper than otherwise. Now we're moving to something else. So this is work I've done for now years with Mike Behrenfeld, which has to do with revisiting Sverdrup critical depth hypothesis. So, which is a cornerstone. It's what we teach our students. I don't know how many. Anybody here took biological oceanography? No, nobody. Well, you might have heard about it. I'm safe. Basically, a very, very nice theory, and it's nice because it's bringing mathematics to biology. The idea is as follows: phytoplankton in the North Atlantic, when there's deep mixing, are light limited. There's too little light for them to grow, and therefore they don't start accumulating. They start accumulating the moment that the photosynthesis here, denoted by light times a constant, is more than a respiration or loss process. That's assumed to be constant. The moment this turns positive, dPdt becomes positive, and phytoplankton accumulate. And this is looked at as in an integrated sense. So photosynthesis is light dependent, and light decays exponentially in the ocean. The respiration is assumed to be constant, and whenever this area here is bigger than what you lose, suddenly you can accumulate. You can start a bloom. So it's a theory about the initiation of the spring bloom. So we just took this float we had and looked whether it's consistent with what Sverdrup said. So here we have measurements surface phytoplankton carbon, and we convert chlorophyll to carbon. There's a standard conversion assuming that the relationship is constant. We can also, which is not true, it varies, but we took an average value, and then you can take also the backscattering and convert it to carbon. And so this is the blue curve, and both of them follow each other pretty well. And right when the mixed layer is maximal, you'll see it reaches a minimum and then start increasing afterward. Totally consistent with Sverdrup, except that Sverdrup did not talk about the surface value. He talked about water column integrated value. And when we look at water column integrated value down to the bottom of the mix layer, what we see is that concentration starts to increase beforehand. About a month before is when we see the initiation of the bloom. Now, why is it in the surface continuing to go down? Because this is concentration. And as the mixed layer deepened, you're diluting the water with stuff from below. So while you're diluting, phytoplankton seems to be starting to increase in concentration. And they do so, this is what we claim, because probably what's happening with this dilution, it's affecting the loss processes. It's affecting the growth processes as well, because as the mixed layer gets deeper, you have less light on average as you circle in the mixed layer. But it seems that the loss processes are affected even more because we're starting to see positive accumulation of phytoplankton in the mixed layer. And that's using two sensors. It brings us back to a story, to a paper from '85 by Evans and Parslow, where they think about blooms as two ways one can look at the bloom. As isolated event, you have a certain condition, the condition happens and suddenly you get a bloom, or you can look at it as part of an annual cycle. And the way you look at it is, if you look at it as one or the other, it makes a big difference, because if it's an isolated event, the condition right now matters. If it's part of an annual cycle, it's really important to know what happened. You don't need to know the initial condition. You can run your model 5 years and then it settles on the right value. And this is important because now you get ecological processes that should come into play here. If loss processes are at work, and loss processes include grazing, it includes viruses, which are unlikely to be a big issue here because concentrations are very low. It includes settling for other reasons of particles. So all of them are playing a role here, and those processes are not part of what Sverdrup had in mind. This is simply how did we calculate, so now we're looking at accumulation rates, or net growth rate, this R. The way we measure it is we have the float going every 5 days, and then in between them we can calculate what was the accumulation or the loss of phytoplankton. We have approximately 6 estimates of growth. What's that? There's something on there. Okay. And we get uncertainties on those because for each month we can get standard error of the mean, and we integrate either to 300 meter or to the max mixed layer and euphoric depth. And these are the distribution of net accumulation rate of phytoplankton that we find. So just so you know, these tail values, 0.05 means that you double the concentration every 14 days. Now, those of you that know something about phytoplankton know that they can double daily, and they do in the ocean double daily. So what this means is that there's a very, very tight control between the loss processes and the growth processes in the ocean. You don't get explosive bloom. Even a spring bloom, it's not an explosive bloom. Things don't double every day. They double in a much, much slower rate. And so it's very hard to look at this process. Even if you look at them in integral sense, this is integrated dB, the rate of accumulation, this is much smaller, this is half of that. This is surface values. And it's about doubling every 14 days. So if we look at this is how the net growth rates look, which are the derivative of the previous floats you see. They start earlier. At the time, they start being positive earlier at the time where the light at the surface is almost the lowest it's ever been, and when the mixed layer gets deeper. And this is not consistent with Sverdrup, again, because according to Sverdrup, in order to get accumulation, you need to improve condition. You need to shallow the mixed layer and get light to them. So now you might ask, and this is consistent with another paper that my collaborator Ben Feld did, looking from space and using models for mixed layer depth. So this was, if you want, the in situ validation of that work he did. So the problem with Sverdrup hypothesis is assuming that this R is constant. Because if this R is constant, the only way you can turn this term positive is by getting better light. It's the only way that you can get accumulation, is if light condition gets better, and therefore you need this restratification of the mixed layer. However, if you add nonlinear terms like, instead of this R, you have something like G times zooplankton, which will represent predation, or you have something like beta p squared, this would be loss to viruses or parasites. Then as concentration change, for example, if zooplankton get diluted, you can get a decrease in the loss rate as mixed layer deepened. So that's the hypothesis we have to what's going on. Again, we think it's consistent also with this issue of the annual cycle that Evans and Parslow talked about. And one thing they said in their paper that resonated with one is this, one awkward conclusion of their work is that the importance of measurement in winter. Now, we don't like to go out in winter into the North Atlantic. And this is exactly where floats are so useful, because profiling floats can be thrown and be there year-round, capturing what's happening in the ocean. That's the beauty of them. So following that work, I wrote a NIOS paper with my collaborator where I pleaded for this kind of float to constrain these nutrient, phytoplankton, zooplankton models, where we'd have a measurement of nutrients. We already have that on float now. We have that kind of, the same kind of sensor for phytoplankton, and something for zooplankton. And I talked today to Tom about getting something to do that. What we need is, and from your community, the acoustic community, is a cheap way to do zooplankton. Something, an acoustic crud sensor that will tell us there's a lot of zooplanktons in the water. No, there's not. It doesn't have to be very accurate. The chlorophyll pherometer is not very accurate, but it's much better than having no information about the predator, the herbivores in the field. And the reason this is so important is because when we don't measure this, we're explaining everything we can with what we do measure. So a lot of what we're doing in the field of oceanography is bottom-up explanation of phytoplankton dynamics. We explain it with light, we explain it with nutrient, because this is what we measure. If we need higher trophic measurements in order to be able to say something about their contribution to what we see in the lower trophic. So this will be a great thing that you guys can contribute to, is this sensor that will provide us. And there are sensors for zooplankton that people did put on floats. The LOPC, the laser optical particle counter, was put on a float, but it's a very expensive instrument. It was done once. And the French are working on putting the underwater video profiler on floats as well. But again, this is going to cost probably $50,000, $60,000. We're not going to be able to equip a big fleet with this. So we do need some broadband acoustics to do that. And again, some information about light and oxygen production would be very useful to constrain photosynthesis even better. Okay, next. So we are moving in this direction, not yet with the zooplankton, but at least with the nutrients and oxygen, pH, and optics on float. There's a big program, $21 million funded by NSF. First time NSF significantly entered the float game. Till now, it's in the US, it's NOAA that is paid for it. And so that's a $21 million project that Jorge Sarmiento at Princeton had. I'm not part of it, but what I did is write a grant for NASA for $600,000 to put these optical sensors on their floats. So this is what I hope to be able to, next time I come here, report about these. They're just getting in the water right now. Just got funded this summer. Another thing we're doing with these is building platforms to calibrate and validate NASA satellites and satellite products. So this is at an UP project where we put some radiometers, both down-looking and upward-looking, to see how well we can use those. These are multispectral. And now we just got funded for another project with hyperspectral radiometric device to try and do calibration for satellite. Now, why is it important? What happened is, when you put a radiometer on a satellite and send it to space, your gains change. They're not what you calibrated it with. This is one. Two, we can only calibrate to within about 1% or 2%, a radiometer. And we need to go down to at least 0.5% to be able to separate the 90% signal of the atmosphere from the 10 signal of the ocean that is measured by the satellite. So in order to do that, we need measurement inside you. And currently we use, the best platform we have is MOBI. It's a NERA-Y, it's a submerged platform, and we get maybe 30 matchup a year with it. So with 30 matchup a year, your uncertainty goes like square root of n. It takes almost a whole year to bring down the uncertainty to where you want it to be with the gains. A year of measurement. That means a year in which you don't trust the satellite. The satellite gains. So if we can at the same time have a platform like this, profiling in the ocean, getting many more matchups in other regions, we can very fast bring the satellite online so that the products can be used within, say, a month or 2 after launch. And this is why NASA is excited about trying to use this platform and has paid for these feasibility studies. And MOBI costs $2 million a year to maintain, one buoy. So again, anything we can do to increase the number of matchup and decrease the cost is of interest to NASA. Going the wrong direction. So to summarize this talk, we're still playing with bottom-up because this is what we have in our measurements list. So if we are to change that, we need many more people to measure zooplankton. The only way it's going to happen is if we have cheap sensors, just like many physical oceanographers now measure chlorophyll on their platforms, even though sometimes they misinterpret the signal they see. And one classic one is called non-photochemical quenching. If you have a lot of sunlight hitting the surface of the ocean where there's phytoplankton, they're going to decrease the amount of fluorescence per cell that's going to come out. So you're going to get, in the surface ocean, a diel cycle of decreasing fluorescence for the same amount of biomass, no change in biomass. I've seen physical oceanographers stand up and say, you can see the daily grazing happening in the surface because of this reduction in fluorescence for chlorophyll. There is no daily grazing at this. I mean, there is, of course, grazing happening all the time, and it varies with zooplankton migration. But that signal of the diel decrease is only it has to do with how chlorophyll is fluoresced by phytoplankton. So it's a physiological problem. It has nothing to do with grazing. It could have nothing to do with grazing. So whatever measurements we do, if it's zooplankton, we have to know something about it, and not just assume that whatever, not try to understand why the signal is what it is. Biogeochemical sensors on floats can provide useful data. I hope I convinced you of that, and can complement remote sensing, because remote sensing only covers the surface ocean and can give us something to integrate to. And finally, and I'm sure you all are convinced, oceanography moves with technology. Every time we put an instrument in the ocean that measures something we never measured before, we learn something about the ocean we never knew before. And so the more we measure with different frequencies, different devices, different sampling strategies, we're going to see the ocean in ways nobody has seen it before. So there's still a lot of room for exploration in oceanography, and I'm sure this crowd is convinced of that, but I'll reinforce that. Thank you very much. Yes, Tom.
You were talking about all the floats, and that's an impressive number of floats that are out there collecting data, and of course there's the observatories that are making a Lagrangian measurement, but fixed in time, fixed in space over time. The other kind of, and I like your floats because it's the sort of force multiplier kind of thing. Have you looked into, I know some people have done this, Tom Rossby did it, but looking at kind of ships of opportunity to put some of these sensors on, particularly acoustic sensors to measure zooplankton or those kind of things to maybe couple in with your float systems. Have you looked at that as a viable way to augment what you're doing in a similar way that you're using this to augment what satellites are giving you?
Yes, so with fancier instrumentation, what we've done is we hooked with a French boat called the Tara that went around the world in 3 years, and everywhere it went, we passed water through hyperspectral spectrophotometer that gives us absorption and scattering in 88 different wavelengths. So it's like a major acoustic multiband. And so, yes, we did that. The boat was free for us. We got some money from NASA to do it. We went all the way around the Arctic last year. We did a circumnavigation of the Arctic, and it was, the data is phenomenal. And yes, you could do it on ferries. It requires system, our system requires, and I was on the boat 3 months out of these 3 years. So it requires that a technician cleans it once a week and change a filter once a week. And that's, the optics is very sensitive to biofouling. So you have to have, and as you know, I don't know how many of you have been on Unal's vessel, I don't know if it's the same on NOAA ships, they often have fluorometer and transmissometer in line, but most often nobody takes care of them. And the values are I hope they don't put it in national database. I'm afraid that they might, because yeah, if you don't take care of the sensor, you get useless data. So it's a great idea, and what Tom is doing is great, but we have to ensure quality. And so as long as we can ensure that, and there are systems, people have come, seakeepers, It's a system, it's a box of sensors, including optical ones, that are put on rich people's yachts. And wherever they go, it measures. And the data is sent to data banks. And then they have people come and replace with a new system, take the old one to get it recalibrated. So there's a lot of work being done with that. I work with French people, again, nonprofits, that put sensors on sailing boats that go around the world. But again, the proof is always in the pudding. Is the data any good? And again, unlike acoustic sensor, optic sensor are much more sensitive to fouling. So whatever we do, we have to make sure that the data is of quality. Yes?
We have a question from Shahak about your NASA grants. What satellites were calibrated using the...
So Shahak, no satellite has yet been calibrated using floats. It's preparatory work for PACE. PACE is a mission that's now due to fly sometime between 220 and 223. And it will be a hyperspectral mission. So very exciting, goes all the way from the UV to the near infrared, many, many more bands than anything we've had to date in ocean color. And that's where our work is now being focused. So what we hope to have is a hyperspectral, we have to build the hyperspectral radiometer. This is work that's done with Atlantic and Wet Labs, and then and Seabird, and put on floats to actually show that we can obtain quality data of sufficient quality to do this calibration.
And a follow-up to that from
Yeah.
him is, for this, is a correlation between a single epoch or an average over time more efficient? Do you have an idea of that yet?
I'm not sure I understand the question.
Well, he asked, what did you find to be more efficient, a correlation to a single epoch of satellite imagery to buoy measurements, or an average over time?
No, they do single
For the
matchup.
no,
Well, the whole time series. Again, because of the square root of n business, and because, again, we know that fluorescence yield per chlorophyll changes in time depending on species, depending on a variety of reasons. And our measurements were done only at night, so we did not suffer from non-photochemical quenching. So you want as much as possible to have as long a record as you can to calibrate against. And then when you treat a whole time series, and that's what we've done. So we just had one number that was based on that calibration, on that cross correlation between the two. And it was to actually calibrate the sensor, not the other way around. We did not calibrate the satellite. But we validated the satellite record in the sense that it was not drifting, and it was consistent with what we measured with the sensor. Yes?
These Argo floats operate in shelf waters as well as the ocean. Do they have, they just drift around, I guess, so is there anything controls their...
So I've seen work by French people. French are very active in the Argo, and what they do is actually they stay on the bottom. They put something on the bottom that makes it such that it doesn't damage the sensor or the float, and they bounce on the bottom in places of 100-meter depth.
So they're distributed sort of equally to the distribution of depths in the ocean?
Oh no, most of them, are designed for open ocean work, 2,000 meter. But you could, the specialized cases where people have, and of course people have found them on beaches too, but mostly it's for global circulation work. Not many people have used it to study shells.
But for the kind of things you're studying, wouldn't the shelves be an important part of the ocean system?
Sure. Yeah. So again, some people, another French group has looked at the Mekong Delta, and they put floats that went to, again, 150 meter in front of the delta to do their measurements. So today it's still a specialized project where you're interested in a specific process that you're trying to quantify. There, as I told you, there's very few floats that as part of the global array currently, and what my community is trying to do is to increase that number, and by showing the utility, what you get out of it, the increased value that these floats have by measuring not just
What's the lifetime of one of those?
now they can go up to 300, 350 profiles. So it depends how often you want to profile. For example, one primary productivity method consists of measuring at 6 o'clock in the morning and 6 o'clock in the evening beam attenuation. and looking at the daily change. And then you have 2 profiles a day. So you're down to half a year if you do 350 profiles. So it depends.
You say you don't recover them? Or?
Yeah, currently they're not recovered. And that's a question again. I mean, once after, if there are enough of them out there, it might be worthwhile to go recover them and have small boats that, when you know that you're reaching the end of the life of a float, to go recover. But when you think about the price of sending a boat to the open ocean to look for them, it varies. A day of a boat is a float with a CTD currently is about $20,000. So there's that. I mean, obviously it pollutes the ocean. You have the batteries are not this kind of stuff you want to have there, but still the ocean is really large compared to the quantity that we're losing large.
That's what everybody says.
Yes.
Yeah.
No, I'm not trying to justify that. I mean, one would hope that one day at least they'll stay floating at the surface when they die, and not get and then they'll drift wherever they'll drift eventually, and people will recover them. At least to do something.
How do you deal with biofouling?
Great question. The reason we did not see biofouling, we think, is because we profiled to 1,000 with this float to 1,000 meters.
You spend most of the time.
And you spend 4 days down there, profile, come down, and the time to telemeter the data has changed a lot. It used to be Argo, so we stayed in the surface order 6 to 10 hours. Nowadays with Iridium, the same amount of data is sent in 20 minutes. So you spend most of your time in very cold, very deep water, and also going through that gradient between the surface and down is not conducive for organisms to grow on. You can, if there is a problem, as they do with seabird, they put little herbicides in them, and you can put also, many of these sensors have the possibility of putting a shutter, a copper shutter, and that's what they do when they're in shallow environments.
I wonder if you could recreate those conditions for your autonomous ship of opportunity system.
Increase the pressure to 1,000 meters. Yeah, sure. But you can blast it with icy waters.
Well, I was going to say it's pressure that'd be expensive, but if it's just cold.
Yeah, that could help too.
Recycling.
The French came up with a system that I don't know why is not yet commercially available, which consists of getting chlorine by hydrolysis of water out of seawater. There's plenty of chlorine in water. And chlorine is what we use in swimming pools to kill pet. And so that's what they use supposedly on packages they have in harbors in France. Ifremer uses that to keep things from growing on it. I'm still looking for the paper, and I'm still looking for somebody who's going to commercialize that technology so that we all can use it. I mean, chlorine is abundant in ocean water. It's just a matter of...
Speaking of extracts, is that spinach extract?
Yes, this is spinach extract in acetone. I've been using it for more than a year, and it's still fluorescing.
So it's in acetone. I thought maybe it was a vegetarian version of cod liver oil or something.
No, not that I know. I wouldn't need it or drink it. Thank you very much.
Thank you.